And I'm going to be talking about the second part, where I introduced in the first part a little bit about protein structure and what is important in it. And now I'm going to talk about protein structure in this part. So let me share my slides with you, and I will begin um, the lecture. So we know that protein structure is very important, and one of the ways by which you can determine protein structure is by protein crystallography. So, and I had mentioned in the last lecture how important crystallography has been in understanding protein structure, because of this, so many Nobel Prizes have been given. Up till 30 were given before, and up till recently also, 2012, we got G protein coupled receptors, which are receptors which help you understand smell, taste, and stuff like that. So because of the importance of crystallography, the protein structure can be determined. And in the last time, I showed you how crystallography was so important, and this is some timeline. I mentioned about Dorothy Hodgkins, but I also said Kendrew was the first one who determined the structure of protein myoglobin, which is like hemoglobin, the protein which makes us our color of our blood red. And these are some of the other structures, lysozyme, insulin I mentioned, which were discovered over time, ribonuclease reductase, chymotrypsin, and so on. And now most of the structures are being deposited in the protein data bank. So this is the place where if you want to go to find crystal structures, you can download them easily, and you can look at how they look structurally. And from structure, with complexes, with various reactants, products, one can also tell about reaction mechanisms. And my next module, I'll show you how using protein structure you can talk about reaction mechanisms. So let me now first tell you about protein structure. So this is an outdated slide with 2012 actually. Now it's 10 years have passed, but so many structures were already deposited and so many proteins were known, more than 9,000 unique structures. So structures in the PDB have been deposited, and you have a lot of structures in the PDB. And only for even one single protein like lysosome, there are more than 1,200 structures. And most structures, they fold into secondary, have secondary structure and tertiary structure. For example, this is an alpha helical protein. This is a all beta sheet protein, and this is a mixed alpha beta protein with a very famous fold called triode isomerase phosphatase fold, which is there, which is this particular fold. And so you can see that they can assemble into secondary structures, and that's what we're going to talk about today. And with time, the number of new folds which are being discovered are decreasing. So what I mean by new fold is how a protein arranges its primary structure into secondary structure, and what is the order in which the helix and the strand it comes. So to understand the structure, I've already told you that to get the picture of the protein structure, X-ray crystallography was used. And we talked about how we use X-rays to collect shine light, which is X-rays, and then we get this diffraction pattern. And we need crystals for amplifying the data. And then we use different sources like the synchrotron to get high energy sources, which are high intensity, tunable, parallel, and high energy resolution we can get. And from there, we get these something called a diffraction pattern. From the diffraction pattern, we are able to collect the data in good detectors, as shown with different qualities, which are shown over here. And from there, we fit these dots to our data, which is made up of something called structure factor. And here, F is the scattering. So more if it's a carbon versus oxygen versus a heavy atom like iron, the scattering will go up. And this is divided into scattering. And you can see here Bragg's plane also come in, because you remember a lot of Bragg's law is used in crystallography. And then there is something which is called the temperature factor, which tells you how much the molecules are moving in the crystal. Because many times what happens is if a region is flexible, then the motion is more and V factor will be high. And it's a function of resolution here. Ultimately, the structure factor is related to electron density. So we convert these dots into which is called a diffraction pattern into electron density, and I talked about it briefly last time. So you will be able to get this wires, which is telling you the electron density. So for oxygen, the electron density will be more because it's heavier as compared to carbon. And this is what it is showing. This is a lysine residue, and how we can then make up the model of the whole protein and into alpha helix and strands and loops as it is supposed to be. And you will be able to see how you can make it. These are different amino acids. I'm going to describe it a little bit later, but you can see this is an aromatic amino acid with a hole, which is called tryptophan. This is another aromatic amino acid with phenylalanine, and this is the backbone of the structure. And you can fit this and then look at the three-dimensional structure of the protein. So when I talk about the three-dimensional structure, let me come to the levels in the protein structure. So there is the primary structure, which is just the polypeptide chain sequence of amino acids. Then there is secondary structure, which I described to you in forms of ordered patterns, which are called helices and sheets, and the unique way in which they come together is called a fold. And then there's tertiary structure, where more than one proteins, they come together. I mean, not more than one protein, sorry, that's quaternary. Tertiary structure, they come together and make some kind of a domain kind of structure, and they arrange themselves. The secondary structures, they arrange themselves in space. It's called tertiary structure. And then when more than one come together, like I said, they are quaternary structures. And then you can get an oligomeric assembly. That would be, sometimes proteins don't work individually, they work as an oligomer. So these are the levels of protein structure, and I'm going to talk about them a little bit in detail. So primary, which is the amino acid sequence. Secondary, like I told you, it can become strands or helices, and then how they come together in closed space is tertiary, and when they all come together or oligomerize, it's called the quaternary structure. For example, this is hemoglobin, and everybody knows there's a lot of allosteric or cross-talk between the monomers in the oligomers, and so quaternary structure is also very important. So lysozyme is a very common structure and one of the first structures that was determined, and it is used in for training purposes when you want to grow crystals, etc. And what the function of lysozyme is, it takes this sugar which are presented in the surface of the cell wall, and it breaks it up and cleaves them, and as a result, the whole cell wall disintegrates, and you are able to lyse the cells. That's what a lysozyme does. So here, what I'm trying to show you is when you study protein structure or you see a picture of a protein, there are very, very ways in which a protein structure is depicted. For example, you can have only the C alpha atom of a protein shown. That means of each amino acid. So then you will see something like this as the picture. So you can see that this is a lot of spaces there, and there's a cleft here. And you can now see a different view, which is called a ribbon drawing, where you can see the secondary structure. So there's a helix, but you really have to peer to see this a helix. But when in a cartoon representation, which is also called ribbon presentation, you can see where the secondary structures are very clearly. Is this a true representation of what the protein looks like? Maybe not. Both of them are not. Because here only C alpha trace is being shown, and here the C alpha trace in the form of a ribbon is being shown. But if you try to get more details in the structure, that means you put ball and stick type of image, then this is what the protein looks like. And then if you put a space filling, which is the real true picture, it looks pretty complicated. You cannot tell anything about the secondary structure here, but you can see a cleft over here. So one thing you need to notice, most of the times where a cleft in the protein or an opening in a protein happens is where the active site is, where the reaction happens. And here you can see this is your substrate, that sugar substrate that we were talking about. It goes in this cleft where it gets cleaved, and that's how the function of lysozyme happens. But excuse me. Out of the representations that are there, it is this representation which you will commonly see in. It's a beautified version, but it is the ball and stick representation, which is the true representation of the protein. But most of the times it's too crowded and too difficult to understand what's going on. So ribbon drawing is used in most cases. Even in ball and stick, you cannot tell anything about what is the secondary structure. But always remember, so these are the different ways in which you can look at your protein structures. Now, since you are going to be looking at protein structures, you should know what are the softwares that people use, and that will be very useful. Suppose you want to start teaching protein structure, visualizing protein structure. PyMol is the software which is to be used. You can have different variations. You can get this in which you can actually look at individual molecules, individual amino acids. You can see tyrosine being shown here. What previously I showed you was the cartoon representation where you can make your strands and helices, and you can even look at electron density images in PyMol. So this is a very popular software, and one should use this software to visualize protein structures. If you want to manipulate density and want to use crystallography more from a point of view of being able to refine your structures, then I would recommend to use something called COOT, and there's a Windows version called WinCOOT. This is the address from which you can download COOT. And if you do this, you will be able to manipulate crystal structures and look at the electron density at different levels and do much more involved crystallography. Okay, so amino acids are chiral, everybody knows, and I have been describing it that they have a C alpha that I mentioned there, and with that they have a side chain which gives the character to it, and I'm sorry, this is the side chain, and this is the carboxylate group, and this is the amino group. Okay, so amino group, carboxylate group, the hydrogen, and the side chain, which is in this case looks like a serine to me, has been shown. Okay. So yeah, serine, as I said. So it can you only have L amino acids, and that's what you have in nature. And so all proteins are composed of L amino acids. Mostly there are about 20 naturally occurring amino acids. There are some unnaturally occurring amino acids also. And the R group determines the property of the amino acid. What I mean by the R group is the side group. All of them have the carboxylate and the NH2 and the hydrogen moiety attached to it, but it is only the side group, which is called the R group, that changes, which is different for all 20 amino acids. Now, the importance of the R group is because it tells you what will be the character of the amino acid. Because you will see in the end, when you form a peptide bond, both the CO minus and NH3 does not exist in this form. They will be linked to another amino acid, and this will be in an amide linkage. So it is a side chain which gives the character of what kind of amino acid it is. So this is alanine, valine. These are all phenylalanine. They're all hydrophobic amino acids. We have the single letter codes also given over here. And sometimes because P is already be taken by proline, this is given as phenylalanine from the voice, the sound that it makes. So methionine, all of them are hydrophobic amino acids. That means they do not have a lot of groups which will be donating making a hydrogen bond, donating electrons, accepting electrons. It will be more like a hydrophobic type of interaction. Then there are also hydrophilic amino acids like serine with the OH group, threonine, and you can see tyrosine, which has an OH group. Phenylalanine does not have, tyrosine has one. And then there are these asparagine with an amide group, and it's this asparagine with two CH2s, longer chain becomes glutamine. And you have histidine, which is a very flexible amino acid because it can get protonated and deprotonated. And I'll talk about the pK. And so it's present in a lot of catalytic sites. Cysteine, a lot of disulfide bond formation, if happens. Cysteine is an important amino acid. And you have tryptophan, which is one of the bulkiest amino acids and difficult to then have very often. So these are some of the hydrophilic amino acids, and they're charged amino acids with head groups such as carboxylate groups, aspartate and glutamate, and arginine, which has got positively charged amino acid, and lysine. So guanidinium group is there, and lysine, but they both have long aliphatic chains, you can see. So they have dual character if you look at it. They have a positive character, charged character at the end, but they do have a hydrophobic character in the middle. So we have these kinds of amino acids which are there, both acidic and basic amino acids. So you can get character from hydrophobic to just having some kind of hydrophilic nature to having charge. Now here I'm showing you the frequency of the amino acid. So if you look here, this is showing you the full amino acid name, the 3-letter code, the single letter code. For again, arginine, because A is taken by alanine, arginine from the second letter has been taken and it is R. The symbol is R. Again, asparagine, you cannot use A, and S has also been taken, so it is N that has been given to asparagine from the sound N. Again, as A has been taken, aspartate is given D. So you can see that single amino acid codes are also very important to know because when you're looking at primary structure, most of the times people express it as single amino acids. Next is given the mass and the volume of each amino acid, and if it's obviously a heavier amino acid, it will have more volume, like 163 you can see for tryptophan, which is the largest amino acid. And again, you can see that now you can see how frequent these amino acids are. If you go through a lot of protein structures, which amino acids you find a lot. You can see the highest number is 11.4, which is glycine, and you can see it's a very small amino acid. You can put it in various places. It has very flexible. It is not even chiral. That's the only amino acid where it is not chiral. And then we have the other small amino acid, alanine, which is there. But some of the amino acids have very low frequency. For example, you can say tryptophan, which is only 1.1%. Why that is the case? Because amino acids such as tryptophan are very hard to make. They are very expensive. So two rings have to be put together. So a lot of biosynthesis is required for it. But there are other amino acids, for example, cysteine and histidine, which are also 1.7 and 1.8. Cysteine is relatively small, but it is a amino acid which is only put by choice. For example, if you want disulfide bonds or you want to have a metal chelator type of thing, and histidine is generally present in active sites, so they are not very common amino acids. So you can see that some bulky amino acids are not common, and then there are some special amino acids which are involved in active sites, which have also been kind of limited and made special. So the pK of the amino acids is also very important because you can see here that some of them have a very high pK and some have very low, but it is histidine which is around 6, and it is the winner here because by a little bit changing the local environment in the protein, you can get a protonated form or deprotonated form. It can work as an acid or a base, and it can be used for catalysis because many of what do the proteins do? Ultimately, most of the proteins are enzymes and they catalyze reactions. So you need amino acids that can be used for catalysis, and to tweak the environment to change the local pK of histidine is the most easy. So it is put in many such cases. So there are other amino acids which are non-standard, like ornithine, homocysteine, gamma aminobutyric acid, and carnitine, and some of them are involved in as neurotransmitters and stuff like that. And these are you know. Homocysteine is not cysteine, but a variant of it. So these are used for special purposes when you require certain specialized functions. But generally, most amino acids like these are not present in proteins because the 20 amino acids take precedence. In the protein, all the amino acids, except the N and the C terminus, which are free, you have an amide linkage. So you don't have NH3 plus or CO minus in the entire structure because everything is taken by an amide bond. So two amino acids are linked by an amide linkage by a dehydration reaction. And this particular reaction is actually done by the ribosome, for which 2009 the Nobel Prize was given. So how to make sure that all the amino acids get added in the right way and the reaction happens. Simple looking reaction, but to put it all together in the right format, it takes a lot of effort. So, so because of the amide linkage that is there, you can see that this particular nitrogen has a lone pair. You can get a double bond here, and CO minus can be there. So you can push the arrows through it. So there's a double bond character. Because of this double bond character, you can see the whole protein has been divided into planes, if you can see here. And this rotation angle between planes are called dihedral angles. And it's these dihedral angles, which is the one between C alpha and the nitrogen is called phi. C alpha and the C O, the amide bond, the other end of the amide bond is psi. And it is the rotation about these planes which is restricted and decided by the R group that is there. So glycine being only having a hydrogen here will have the best rotation. Otherwise, if you try to rotate the bonds too much, you may have a clash between the R groups that can happen. And you can see they are also located opposite to each other so that the clashing is less. But this 5 psi angles were analyzed by someone called Ramachandran. He's a very famous guy. And they are called the torsion angles, like I explained to you, and the definition. And what you can do is you can rotate in one direction 180 degree and you can rotate in the other direction 180 degrees. So you will have plus 180 to minus 180, and that is what is represented on Ramachandran plot, who theoretically went through structures and predicted that structures, when they form some kind of arrangement, they can only proteins and amino acids, when they form some kind of arrangement, can only take certain preferred 5 side angles. And this whole thing has been developed because of this rigidity in the double bond character that has been introduced by the amide linkage that is there. So they are restricted by the 2R groups, like I said. Okay. So Ramachandran is the person who did this, and if you can see in the helix, it is wound up in such a way that there are certain regions in which helixes are there. So let me explain this diagram to you. So this is called a Ramachandran plot, and you can see that again, psi angle 180 minus 180. So minus 180 to plus 180 is 360 degrees, and minus 180 to plus 180 is again 360 degrees. So basically, rotation this way, completed to 360 degrees in both ways. It's a rotation of the planes. So the planes, the 5 psi angle, there is a region, extended region near plus 180 and minus 80 is for beta sheet because it's extended, and it's more bound up around this region around 50 minus 50 to plus 50 for the helix, because you can see that the helix has these turns and there is a definite torsion angle that comes with it. So most of the helices are right angle helices, and most of your structures you will see in this and this region. If you have a protein structure where most angles are in this region, you know that your structure is correct. So Ramachandran's plot is used to assess the quality of a structure in that way. All right. So this is just a styrofoam model which Perutz made when he saw the structure of the hemoglobin, and that's how they used to do many years ago. But now we do everything in the computer. And I already talked about it. So let me skip that slide and go. When you go from primary to secondary structure and secondary to tertiary structure, what holds the structure together are hydrogen bonds. So hydrogen bonds in protein are very important. So you will have a donor and an acceptor. So that's what it is written, donor and acceptor. And between them, you will have a hydrogen which is shared. Okay, so bond angle is generally 180 degree in most cases, and it is used to stabilize the secondary structure. So you can see that these are the different distances, and let me only show the donor acceptor distance here. That is what we look at in crystallography because we cannot see the hydrogens. They're too light for electron density to appear. So you can see that that you can have oxygen, nitrogen, oxygen, and distances are around 2.8 to 3.4. That is the limit between the acceptor and the donor that you can kind of see in a crystal structure. So in crystal structure, there will be no hydrogen, so you will only be looking at acceptor and donor distances, and these are some distances to keep in mind to tell whether a hydrogen bond is there or not. So when these hydrogen bonds will come together, we will form these type of structures, and alpha helix is the first structure that I'm showing you. It has 3.6 residues per turn, and the height is around 5.4 angstroms when you complete this whole turn. And there's a rise of 1.5 angstrom and a rotation of 100 degree for each amino acid to be as it is present in the helix. So the width is around 5 angstrom. So let me show you an alpha helix, which is generally alpha helices can be 12 to 15 residues long, and you can have many alpha helices. And I have already told you its turn contains 3.6 residues and 100 degree per turn when viewed along the helix axis. And I'll show you alpha helices with L amino acids are right-handed. Okay. helices located at protein surface are usually amphipathic, and I'll describe to you what it means. Amphipathic means it has both hydrophobic and hydrophilic character. And those who are inside the structure are very hydrophobic, means amino acids, the side chain character would be of hydrophobic amino acids like valine, leucine, etc. So when you look at the helix axis down, like here, so if you were to look along the length, the helix will look like this, and then you turn it and you look, you will only see the open face of the helix. And you look down and it will go all the way down. And that's what this is. These are the C alphas, and then you can see all the amino acids have been arranged. And now let me show you how this is the first amino acid, and this is 100 degree away the second amino acid, and another 100 degree away the third amino acid and the fourth. And then you change your turn and you go backwards and you can look at the projection. Every 100 degree there will be an amino acid, and this is called a helical, this type of representation is called a helical wheel representation. So let's look at amphipathic helix, and we'll actually put now amino acids. So first amino acid being alanine, second being threonine, then you have the third amino acid being serine, then leucine again, and then you have tyrosine, then you have arginine, and then you have alanine. And now you, the last one being alanine again, and second last being alanine, and then you have glutamic acid and serine. So you can see that now if I were to divide a line and put it through, you will see all these amino acids are hydrophobic, leucine, alanine. But in this side, most of the amino acids, serine, OH character, arginine positively charged, serine, glutamic acid, tyrosine with an OH, threonine with an OH are all some kind of hydrophilic or charged amino acids. And this kind of division between helix, this side facing the inside of the protein, this side facing the solvent, is called an amphipathic helix. So hydrophilic edge, hydrophobic edge inside the protein structure with other helices outside to the solvent, and called amphipathic helices. So this is again a picture of a alpha helix being shown with a very nice electron density, and you can see there's a hydrogen bonding pattern which is there, where the backbone is hydrogen bonding, and the pattern that you will see is I plus 4. Okay, that is the pattern that you will be observing. That means between the ith and the I plus 4th atom there is a hydrogen bond. That is how the hydrogen bonding is described. So you can have I, like I said, then you have 2, 3, 4. This is your I, and the amino acid is C, and the C double bond O. And then you can see that the second amino acid, the third amino acid, and the fourth amino acid is when I and I plus 4 is what the hydrogen bond is through. Okay, so this is the characteristic of an alpha helix. Between the I and I plus 4th residue, you will have a hydrogen bond. Such things do not happen in sheets because they are flat, not really flat, they're like asbestos sheets, if you remember, but they are more extended structures. And so they have a different different way in which this play out. And you can see here, you can have sheets where you have the amide nitrogen of one with the C double bond O of another, hydrogen bonding. And you can have one strand going in one direction, the other going in the other. They are called antiparallel. And you can also have parallel strands, but this is just showing the hydrogen bonding for the antiparallel. And you can see the antiparallel, how the C alphas are very close to each other, and more nice looking one. And this is more kiltered, and this is parallel, where both of the chains are going in the same direction. And you can see N to C, N to C, N to C, and in the other one it was N to C, and you can see N to C. They were antiparallel. So it looks less kiltered, but ultimately there is no such thing that parallel or anti-parallel sheets are preferred. Both have equal preferences and stability is the same. So one is not better than the other, but yes, by looking at it, one looks a little bit better than the other. So you have both parallel and anti-parallel ones, and you can see that this hydrogen bonding is there. If you call this I plus 1, I plus 2, you will have I to J, and also then you have I plus 1 to J plus 2 and stuff like that. But it is more like a side-by-side thing that is happening over here. And you can see that in the parallel, you'll have J, I plus 1, and so on. And ultimately they are called beta pleated sheets because you will have pleats like sari pleats are there. There are pleats that are formed. It's not a flat structure. I showed you that, like the asbestos sheet. So they are not like, sheet doesn't mean completely flat. It means that it is more like a corroborated structure that is coming up, and that's what you're going to get something like pleats that are going to be there. And ultimately, they will not be just flat structures. You will see that there is going to be a spiral thing that is happening. You can see they are just forming a spiral, and then there is helices on the outside, or they can form barrels that is forming. So it's not like a flat structure that you will get because you have a sheet always. Now, between the sheets and between the helices to connectors are the turns. So here you can see there is this I plus 3 type of turn. That means the first and the third amino acids are hydrogen bonding via the backbone. And you can see that the turn can be different types, type 1 and type 2. And that depends on whether this C double bond is outside or C double bonds who is inside. And these turns are the ones which give flexibility. End of the structure, you will have these turns, and then you can have loops connecting secondary structures. And they are very important items that are or important in changing the structure, direction, in going from helix to strand, and act as connectors. There is another kind of helix called the 310 helix, and also there is something called pi helix. But the 310 helix, you can see, is a right-handed helix with 3 residues per turn instead of 3.6 that we are used to. So 310 helix is more compact helix. It will be much more compact, and they are generally short. And you can see this that if alpha helix has this much space, when you look down, 310 is much more narrower because it's a I plus 3 type of hydrogen bonding pattern. So when we look at these amino acids, we talked about the secondary structure, etc. We want to know which of the amino acids they appear a lot in secondary structures like the helices versus beta strand and versus turns. So you can see this is a collection. Whenever you have a number of, it's a probability being shown, greater than 1, that means it is much more probable. Less than 1, not so probable. So glutamic acid, for example, prefers being in an alpha helix, not much in the beta sheet, and in reverse turn also it's okay. Okay, now you can look at this methionine residue. It likes to be in alpha helix and beta strands, pretty much. Little preference for alpha helix, not in turns, because the probability is quite low for it. There are some amino acids like you can look at valine, isoleucine. They seem to have a higher propensity towards being in a beta strand. So you can guess what kind. If suppose you have a primary structure, if you have a collection of sequence, you can tell, okay, this must be for a beta sheet type structure because it has valine, isoleucine, tyrosine, cysteine, etc. And this column, this type of amino acids, which propensity above 1, prefer being in an alpha helix. And there are also these type of amino acids which prefer to be in a turn. Glycine, not surprising at all, 1.77, much greater than 1, because it's very flexible. Proline is also considered to be a helix breaker, for example. It is at a tip of helices. It's also preferring. So you can see some of these amino acids, they want to also prefer themselves to be always present in turns. So from here, you can kind of get an idea of what your sequence you expect to be and what amino acids will be present in what type of secondary structure. Okay, I already mentioned this for you for the 3 different sets. But ultimately, these all helices and strands, they come together and form something called a tertiary structure. So they're connected to each other, and the way in which they come together, like for example, you can see here it is showing 1, and after 1 there is a helix. Only beta strands have been labeled here, 2, and then 3. And then the whole structure goes far away and 4 comes in. So 1 and 4 are close to each other. And then you have 4 close to 5 and 5 close to 6. And this type of arrangement, when you put it in 2D, it is called a topology diagram. So you can see this is a topology diagram. It will tell you in a flat diagram which elements are close to each other and which are far away. So because all the time we cannot visualize by pi-mole or not, but we can draw these kinds of diagrams and they're called topology diagrams. So here 1 and 4 are close to each other. You can see the same thing in a topology diagram. And whether the helices are parallel or anti-parallel, you can also see that all helices are parallel in this particular Rossmann. This fold is called Rossmann fold structure. Okay, so you can have a structure which is all alpha helical, all beta sheet, mixture of alpha and beta, or so. For example, this is alpha beta structure. You can see the sheets are the sheet is formed in the middle by strands, and then there are helices on the outside. But the arrangement of all of these being sheet in the middle and helices on the side, the way in which the sheets are arranged are different. So order is 1 is close to 3, 3 is close to 2, like I told you in the topology diagram. So these are different folds that they are put into, and the first protein which was found in this fold, or a popular protein, is by which the fold is named. For example, P-loop hydrolase. And remember the previous image I had told you, this is the Rossmann fold put again here in this arrangement. And these are all alpha beta folds, because you can see there is this is called a Tim Barry fold, very famous. Okay. So proteins can not only do this, they can have one protein can have multiple folds. This is all beta sheet fold. These are two all helices. So they can have different domains, like pyruvate kinase here has 3 different folds coming together. They are more complicated in nature. Okay, so N-terminal barrel domain, alpha beta barrel domain in the center, and C-terminal you have the helical domain. Now, again, a quaternary structure is when more than one tertiary structure comes together. So you can have oligomers. They can assemble, the monomers can assemble as quaternary structures. And these are example of a dimer, a trimer, a tetramer, in which they can be in the same plane, or they can be not in the same plane. Then we have a pentamer. Then you have a planar hexamer where there's 6 of them. And you can have a hexamer where 2 of the trimers are like a sandwich. One is on the top and one is on the bottom. And you can have more complicated structures keep on going, like this very different layers which are being put into it. And so proteins can be very complicated to very simple, and they can assemble as oligomers according to what type of function they want to do, and they may become more complicated. Icosahedron is also shown here, where viruses take this up. So this kind of structures can exist. And with this, let me end my lecture here today. And I'm going to again stop sharing my screen and go to my next module, which will be soon to come.